For this session, we're going to be talking about how AI is reshaping biotech. No surprise there. Dr. Nick, of course, stays on with us. And we welcome Dr. George Sledge, who is with Caris Life Sciences. And in his role, he oversees medical affairs, research, and medical education, including oversight and leadership for the Caris Precision Oncology Alliance, as well as Caris's global team of medical science liaisons. Dr. George Sledge, good to have you in the house. I'm going to turn this over to Dr. Nick, and you guys can take it away.
Fantastic. Thanks, Carol. Thanks for everybody for hanging on. Apologies for the difficulties. George, thank you for joining us. I did provide a little bit of an intro when we were waiting earlier, and I sort of tried to put some context around it. Obviously, completely did a terrible job, total injustice in terms of the
I can't correct you since I wasn't on.
well,
So, so don't worry.
unfortunately, everybody on this call can, and there are some questions coming in which we'll get to as best as we can. But thank you for getting on. Sorry that there was problems.
It can't be any worse than when I asked a particular AI program to provide my CV a week ago. And I learned to my great interest that I had graduated from the University of Tennessee Medical School, which makes me somewhat cynical about AI and medicine.
Yes, we're well worn out phrase of hallucinations comes to mind. I think I've set things up. Hopefully, people have that. So AI in biotech, and we're talking about that specifically. This is super exciting in terms of the opportunity. I think Caris is at the forefront of some of this, and what I'm hoping you can do is share a little bit of your perspective on how AI is making a measurable impact. And I just to be clear, I did share a little bit of a teaser on your story on the breast cancer that you had discussed. So, but I think you can add so much more. So please tell us why you're excited, aside from your CV, obviously.
Well, we're excited because basically for the first time in human history, we have data sets large enough to answer big questions in ways that we weren't capable of doing so before. And we're capable of doing that because of artificial intelligence, because the ability to apply neural networks to these large data sets and the ability to bring multiple data sets together at once. So for instance, at Caris, and we're not unique in this, we have over 500,000 patients worth of whole exomes, over 500,000 patients worth of whole transcriptomes. We have digital pathology, where we've digitized literally millions of slides, some H&E, some immunohistochemistry. And we have the data scientists who are able to take all of these data inflows and attach them to clinical data that we get from claims data and electronic health records. And when you're able to do that, you of course are able to ask lots of questions that we wouldn't have been able to ask previously. But my career as a researcher was based around phase 3 trials, very large clinical trials, but those trials are good for answering really big questions and not good for answering a million other smaller questions. Though we still are very interested in the phase 3 trials because, as you've already discussed, being able to apply modern artificial intelligence to those trials has allowed us to answer old questions in very new ways using very exciting technology. So it's a fun time to be a cancer researcher, and it's a particular fun time to be able to apply these new tools to the old problems.
So, I mean, just to be clear, I want to be sure I'm understanding this correctly. What you're saying is that the data's been there. We weren't able to analyze it in a way that was actually helpful to us. And now that we can bring this together in a more meaningful way, and I think importantly, you said apply live clinical data, we're starting to get new and revolutionary insights.
Indeed, indeed. For instance, I mean, when you have 500,000 whole exomes, 500,000 whole transcriptomes, think in terms of what that actually means. So an exome is 23,000 genes. It's all the genes in a cell. The transcriptome is 61,000 transcripts per cell. So you're applying that in turn to tens or hundreds of thousands of patients, and you're applying it to their clinical data, which in turn may be similarly complex. And then you're also now applying that to images, which can be very, very complex. And again, the old problems that we used to address. I remember my first clinical trial that I ever published in the Journal of Clinical Oncology was a phase 2 trial with 20 patients. The data sets that I deal with now are 2 or 3 orders of magnitude larger than that, probably 3 orders of magnitude larger than that. And that just allows you to answer really cool questions in a really cool way. I mean, just to give a very simple, for instance, at the ASCO meetings on Monday, I presented some of our own data where we were looking at a kind of unusual problem in breast cancer, which is to say ESR1 amplification of the estrogen receptor gene. We had to go through 30,000 breast cancer patients to find, I believe it was somewhere around 120 patients who had amplification. And so just being able to dig into those large data sets allows you to do things that you wouldn't have been able to look at previously.
I, Fantastic. So, I mean, from your perspective, you've been at this for an extended period of time. You've seen lots of variation and, I guess, increasing numbers of patients,
Yes.
for a variety of reasons. But now the ability to use that data and process it. Can you give us some real world examples of some of the insights, the utility that you've derived as a clinician?
Well, sure. Let me talk about what we've been doing recently with a product we call MyClarity. MyClarity is based around digital pathology.
Hmm.
Uh, One of the things that's always fascinated me is, if a pathologist looks through a microscope at a infiltrating lobular carcinoma, that pathologist can identify that literally in seconds in many cases. Um, Little known to most people is that just the very identification of ILC, classic pattern recognition by the human brain, allows you to identify a single gene mutation in the e-cadherin gene. Now, if that's true for the human brain, the human eye, imagine what it's like for artificial intelligence, which is now able to look at very significant and misunderstood or totally not understood at all patterns that are present in a slide. You take a hematoxylin and eosin slide, cutting edge 1877 technology, the standard slide used around the world by pathologists. And that slide has immense amounts of data. It has data that tells you about the immune system. It has data, as I mentioned, that may tell you about mutations in the cancer. It has data about proliferation. It has data that we can identify off of a slide that tells you whether or not there's microsatellite instability in that patient's tumor. Uh, Even beyond that now, if we take those in bulk, and we've done this in the form of a number of large phase 3 clinical trials, you can ask the machine to identify the patterns that are correlated with particular outcomes. So, for instance, in the analysis that we've done recently, we can take patients with early stage estrogen receptor positive breast cancer. These are patients who are in some cases, at heightened risk for an early recurrence due to rapid proliferation of the cancer. In some cases, due to a very late recurrence that may be 10, 15, 20 years after diagnosis. And often off of an H&E slide, we can actually tell you who's at risk for early recurrence and who's at risk for late recurrence. Again, based upon our ability to apply artificial intelligence and to tease out from these individual slides what's going on. In turn, we can take those slides, and because we've done next generation sequencing, looking at the transcriptome and the exome, we can apply the knowledge that's obtained from those to the slide. This now gives us the ability, and this just, Nick, this just blows my mind. We can have someone send us a slide, take a picture of a slide, digitize it, upload it to Amazon Web Services, send it to us. We can apply an algorithm to it, we can turn that around within 24 hours and send back a result that tells whether or not a patient's at high risk for early recurrence, in which case you might need chemotherapy, or at high risk for late recurrence, in which case she'll need extended adjuvant hormonal therapy. This is something that we just simply couldn't do,
Mm.
you know, even 2 years ago. So, so the ability to apply artificial intelligence to these large data sets is allowing us to answer really big, really important questions that directly impact patient care.
So, I mean, it reminds me a lot of astronomy, where we're re-observing some of the data that we gathered primarily from non-visual range, and
Mhm.
finding things that now confirm new insights and so forth. And much of this is about seeing more, and also the ability to bring it
Mhm.
together. Um, I, you're clearly you're using it, you're practicing,
Yep.
it's giving you value. How we integrate, I mean, it always, a lot of the time for me, it boils down to workflow and integration. And I'll try and tease in a question that came up in the chat, which talked about security, that they intimated that maybe it's AI agents if you're using AI agents. And the question was specifically, how do you ensure the security and trust if that's what you're doing? So I know that's a big question. I've sort of lumped in something, but how is this all working in the flow, and how's the security?
So it's a great question. I guess the first thing to say is that you know companies such as mine have been dealing with this issue for a very, very long time. I mean, HIPAA rules have been around for a long time, and we get punished severely if we violate them. So we have large banks of people whose sole job is to take care of that sort of level of security at a level that I certainly don't understand. Um, I think beyond that, when you are applying artificial intelligence, for instance, certainly to the human genome, there's always a concern as to whether or not you can identify an individual off of that particular DNA.
Mm,
But this is a matter of keeping that data just really highly, ultra secure. Beyond that, I would say is that while we use individual patient information to develop signatures, to develop algorithms, we don't then go back to those patients when we're looking at a new patient. So if for instance, in the case that I just mentioned with the H&E slide, uh, someone sends us a picture of an H&E slide, there's no way that I'm aware of anyway that you can identify an individual off of a
right.
hematoxylin eosin slide
Yeah.
that's sent over the internet. So I'm while I share the concerns if we were talking about moving large DNA amounts across the internet, I'm less concerned for many of the other applications.
Right. And then from a workflow standpoint, how is this integrated into the practice? What do you, how are you and your organization struggling with this?
Yeah. Well, so we struggle with this in lots of different ways, as I'm sure your other speakers have mentioned to you. Uh, the very simple matter of having the electronic health record attached to external vendors, can be a significant issue on a regular basis. But we have teams that do nothing but that. So that just becomes part of our standard workflow. For things such as what I mentioned, again, this particular what we call my clarity, the ability to use NGS and digital pathology to answer these sort of questions. This is actually fairly simple workflow, and that is to
Mm,
say, uh, the doctor doesn't care about the algorithm. The doctor cares about the result. So the workflow is fairly simple. The doctor calls up his pathologist, says, send some slides to Caris. Caris processes them internally, sends the result back. It goes into
mm.
the electronic health record. So that part I think is fairly straightforward.
And relatively, I'm going to say new technology. I know we've been seeing a lot of this, but it is still new. And to be clear, you and I grew up in a healthcare system that, you know, we knew the answers, this sort of font of all knowledge, and clearly
Mhm.
with the extent of medicine, that's not true. And I'm just sort of setting the stage, but what I'm primarily thinking about is what's the reception of this? Because if you come back and you come back with something quite different to what the clinician is thinking, is there an accept? Do people see this as a real positive? How's that going?
Well, I always tell people that the hardest job in the planet right now is to be a general medical oncologist. And I say that because if you are a general medical oncologist, if you go to the FDA's hematology and oncology website and look at new indications, there's literally one new indication every week of the year for the
Wow.
past 5 or 6 years. And so I'm not
To be
smarter.
clear, I want to be sure that everybody understands that. New indication for treatment of a disease with
Yes,
a therapy of some description.
yes,
Yeah.
indeed. So think of now, new indication is not a new drug because a new drug may be used for
Right?
several indications. But think about that. Think about what that means. If you are a general medical oncologist and you are in clinic and you're seeing a breast cancer patient, a
Mm,
colon cancer patient, a myeloma patient back to back, and every week there's something new
right.
uh, that where you have to learn about toxicity of therapy, where you have to learn about where it fits in the larger framework of the treatment, where you have to worry about all of the insurance stuff related to that because the United Healthcares of the world may not be friendly in terms of rapid access for expensive new drugs. It's a really tough job. And so, again, part of what's going on in the AI field right now in the clinic, of course, is how do you keep up to date on these new therapies? I'm sure you've heard of open evidence, which
Mm.
I would say has become a
Frequent user.
yeah, and has become a leader in this area,
Yeah.
so that certainly remains a significant help, and I'm sure there'll be others coming along as well. Uh, Clinical decision support, that's offered through the electronic health record, I think is going to become increasingly, important here in this era. At the same time, that's just the drugs. Of course, there's also the diagnostic issues, like the algorithms that I've mentioned. Uh, Physicians are used to getting streams of knowledge from multiple different angles. They have to be. I mean, the average
Right.
physician, lives, in a torrent of chaos, throughout the day. If you're seeing 20, 25 patients and
Yep.
nurses are circling
7 minutes
around.
apiece, and yep.
Yeah, so that's part of our normal lives. But just the sheer increase in the number of therapeutics and the number of diagnostics has made that job ever harder.
Right.
Mhm.
Um, so I think, is it fair to say you're successfully implementing this in your own clinical practice? Is that
Mhm.
true?
Yeah,
So
I'm yes. Mhm.
as you think about that, so you've done that, your institution has done that.
Mhm.
I think one of the pushbacks we get from a lot of this AI is this pilotitis, or too many pilots. And how do you get from there to actual utility where people are going, they barely even notice that they've got a new tool? What separates pilot from success, do you think?
Mhm. Well, within institutions, of course, there are ways of simplifying things like this. Um, I was when I was at ASCO the other day. I was speaking to the leader of a large uh, private cancer group in Los Angeles, and I asked exactly this question: how do you solve this problem? How do you do it? And first off, he pointed to the importance of guidelines. Uh, Organizations such as the NCCN and ASCO and ESMO and others, regularly update guidelines. These guidelines have made some things simpler for physicians. There is always the danger there, however, with a guideline-based practice that you are in essence practicing, cookbook medicine that may not be particularly well individualized. So I think that that's a continuing concern. But just from a sheer survival standpoint, uh, being able to have a ready reference that you can go
Mm.
to that will, that will give you, what is last week's treatment from ASCO as an update, becomes very, very important and indeed crucial for survival if you're a doc.
Interesting. Question online, and I think this is it's tough, but I think it's important to sort of address it, think about it. There's increasing pricing pressures, for all of these new biotechs and the cost of these drugs and so forth. And as I think about this, there's, you talked about it, reuse, reallocation. Um, there's some fear, certainly coming from the audience here, that this potentially offsets and might decrease some of the innovation because now there's not as much of an incentive. Do you see that as a valid
Yeah,
concern?
it's not just fear, it's a reality. I mean,
Mm,
healthcare is either the leading or second leading cause of people going bankrupt in the United States.
yeah,
I mean, these expenses are ridiculous and ridiculously high. My feeling there, and again, the one new indication a week certainly magnifies the economic burden.
right,
I mean, all of, when you're talking about drugs that, are costing 10, 15, $20,000 a month, then you're talking about an astonishing economic burden where most families just simply can't afford it,
right.
if they're not adequately insured. So, the fixes there, of course, are the economic ones, the fixes related to things like Medicaid expansion and the like, that are important. But beyond that, the science fix is an interesting one. Uh, we talk about precision medicine. Most of these new drugs, even though they are precise in that they have a specific molecular target, are not particularly precise in terms of telling you who's going to respond,
Right.
who's going to
Who
benefit,
who should get it versus who shouldn't?
who should get it, who shouldn't get it. And so part of what's exciting about this AI revolution that we're going through right now is that because we're allowed to look at claims data, electronic health record data, attach it to all this molecular data, um, we can in a really, really special way predict which patients will or will not
Hmm.
respond to many, many
So getting
therapies.
better value for our spend, right?
Yes, I mean, first off, the best way to avoid toxicity is to not take a drug that won't work, right? I mean, that is absolutely the best way to reduce healthcare cost related to toxicity.
Hmm.
If we know a drug's going to be ineffectual, then you shouldn't give it. And I'm actually confident that the AI is going to be a major part of this
Hmm.
solution. There are barriers there. I've always, I've posited this to, in discussions. I would love to have access to many large databases that exist within many pharma companies. But I imagine the chief medical officer for Merck or BMS or whatever going into their boss and saying, George Sledge would like to develop a signature that will tell you the 50% of the patients getting our $32 billion a year drug that they'll benefit or not benefit. I would imagine that CMO would be looking for a new job in a hurry.
Mm.
Uh, and so I think part of the fix there that we're going to need is actually to make sure not only that we have great new drugs, and congrats to the companies for coming up with them, but that we're able then to use our other data and use our novel artificial intelligence technologies to identify the patients more or less likely to
Right.
benefit.
Well,
Great.
I think great insights. I'm going to leave you with one quick question. We're out of time,
Mhm.
and it actually came from the audience, and I think it's a great closer for you. What's the future of this AI technology for cancer treatment? Where's you're excited now, but I'm sure you can see, and I will ask you to be brief, please.
The data sets will get larger and larger and larger. I said we had a 500,000 of this and a 500,000 of that. 5 years from now, that will be millions and millions. the data will come in quicker. So we'll have larger answers quicker and better answers for patients. I think that will change things in a hurry. That will allow us to say, Mrs. Jones, you have breast cancer type 3B. We know that your cancer will respond to this drug. We know that based upon our ability to do next generation sequencing on circulating tumor DNA, that we'll be able to predict when a resistance mechanism will occur, and then we'll be able, based upon that data coming out of that analysis, to tell you which drug you should take to prevent progression. That's an exciting future, and
Great.
we're very close.
It
Exciting
is.
stuff. Dr. Sledge, thank you so much.
Thank you,
It's
Dr.
a pleasure.
Sledge.
I'm going to hand it back to Carol.